So for those people who have gone through single molecule FRET, it'll be a good review plus a lot of more materials. Okay, so can you hear me in the back? Okay? Okay, so I just kept my main title because this is the title of the main story, but I have some more materials to introduce the topic. So, but anytime if you have questions, you can raise your hand. Feel free to raise questions. Okay, so all right. So I have this thing. I've seen a talk where they gave this kind of thing, so you can see the progression of the talk, and like by this time you'll be glad that more than about half of the talk is over. So hey, there's another one I know. Okay, so we do single molecule fluorescence imaging, and you probably heard about FRET, and there's two-color FRET, but it could be a multicolor FRET as well. So why do we, try to do studies at single molecule level? If you think of biology like a huge orchestra playing a grand music piece together, while we can listen to the entire music and enjoy the flow of the music, sometimes we get a greater insight by zooming into a single player and looking at his melody. Sometimes you can look at you can zoom in to just listen to two people's lines together in duo. Yeah, lots of people I know. And you can learn about their role, the role that they play in this music, the role that they contribute to this music. So yeah, so he enters first in this case, and then she enters a little bit later. He goes into this crescendo mode, and she enters in this dolce mode. So there's this dynamic, loud and soft, and there's an interdynamic between the two molecules, as well as their interactions that contributes to the entire music. So that's somewhat like our single molecule measurements. We look at molecules in isolation. So there are different ways that we carry out the single molecule fluorescence detection. So in terms of colors, if you didn't know, we can use just one color to detect single molecules. Sometimes we can use two colors, and this is the well-known FRET, fluorescence resonance energy transfer. And this can be done at 3 colors or 4 colors even. So if you find the right donor and acceptor molecules that have the optical properties that allows you to do energy transfer, you can do up to 4 colors. Maybe you can imagine 5 colors in the future. In terms of the way that we monitor signals from these molecules, sometimes we immobilize substrate, such as DNA or RNA, could be a protein as well, and then a partner molecule. In this case, a DNA or RNA interacting protein can be added. Okay. Sometimes we can immobilize protein and then add substrate to take measurements. There's this new method called single molecule pull down, which allows you to pull down molecules directly from cell lysate by antibody specificity. There you can do more complex studies such as stoichiometry. I don't know if you're familiar with this term, but if you're wondering about, the beauty of this assay is that the molecules are, the native context of the molecule is preserved because you pull out molecules directly from the cell lysate. And this works really well for good antibodies. So these are different ways that we immobilize molecules and take measurements. Okay, so review about FRET. So FRET is nicknamed the spectroscopic ruler because you can measure distances by looking at intensities of a pair of dyes. So this is a FRET this is the energy transfer efficiency that goes from 0 to 1. And so this is a FRET sensitivity curve, or distance sensitivity. And this is the distance in angstrom. So FRET, so when you have a pair of molecule, green one I'll refer to as donor, and then the red one is the acceptor. When they're close in distance, you get a very high energy transfer. As their distance become farther and farther apart, you get lower and lower energy transfer. So that's as simple as it is. So for example, you cannot tell if you're wanting to monitor a distance change between like 10 and 20 angstroms, we cannot do it. But you can This is the Fred-sensitive range, roughly about 25 to 75 angstrom distance, or 2.5 to 7.5 nanometer, which makes it an ideal tool for probing biomolecular interactions. Okay. So on the slide, we use a quartz light, which is silanized. Then we attach this molecule, coating molecule called PEG, polyethylene glycol. This is an important brush layer for rejection of any non-specific protein binding. So this takes a whole day to prepare the PEG slide, but it is crucial for all the measurements that we do that involves protein. And then a small percent of the PEG, 1 to 2% of the PEG, is biotinylated such that we can layer it with streptavidin. Then we can attach biotinylated molecule of interest, such as biotinylated DNA or biotinylated proteins or biotinylated antibodies. So after attaching a molecule of interest, in this case I'm depicting a donor-labeled DNA. We can flow in, for example, acceptor-labeled DNA. Then you can flow in the donor-labeled protein. When this is ready, we shine it with illuminate a small portion of the slide with the laser, which creates an evanescent field. And this is the thin, narrow strip of region that we actually collect intensities of donor and acceptor. So it doesn't matter if you have too much protein, as long as it doesn't get into the evanescent field area, your noise rejection is done automatically. So we only detect molecules that are very close to surface, i.e. the molecules of proteins that interact with the substrate. Okay, so brief overview about experimental setup. It is simplified. So here is our reaction chamber, and prism sits on the top. We use the prism type for various reasons. If you're interested, I can tell you more about it. So laser is coming in this way, going out this way, and this is where the evanescent field is formed. Then quartz light sits on top of the platform. Then we have the objective to collect the light. Then it goes through filters and lenses, and there's a dichroic beam splitter that splits the donor and the acceptor beam. And we recombine donor and the acceptor images side by side such that we can monitor the donor and acceptor intensities side by side. And you can see that the molecules are mapped out nicely because they come from the same set of molecules. Does that make sense? So we have, probably about 10 different DNAs possessing donor and acceptor, and we can see that they are exactly in the same position. It's just we're looking at it in two different screens, one for donor and one for acceptor. Then we can, when we collect the intensities, we can use this equation to calculate the Fred efficiency. Okay, so using single molecule Fred, we've done a lot of interesting work. I just wanted to show you this because many of you have seen this in your class, if you took my class. So in this case, it was a translocase or a motor protein that we knew that would walk on single-stranded DNA. So we labeled it with a green dye in a single position, then subjected it to red-labeled DNA. And very unexpectedly, this protein showed this kind of repetitive motion. So what you're seeing here is the intensity fluctuation between the donor and the acceptor. And what you can interpret, the interpretation we made was that protein travels this way, hops back to the 3', and then travels this way, and it repeats its motion many times until the protein dissociates or the dye is photobleached. So these are the intensity profiles we get, well, the raw data we get, and then we calculate the Fred efficiency based on the intensities. But it was here, as you can see, I'm moving on to the next topic. So this is what we can do with Fred. It's a lot of fun. In the microscope room, basically you're seeing this kind of seesawing sets of molecules everywhere, and it's very much like a planetarium kind of experience because you're looking at fluorescence going back and forth. So in this experiment, obviously this isn't the only experiment we did. We played around with the dye positions and such. And we learned soon enough that, so this publication was 2005, but already in 2004, we saw that, the exactly same motion can be visualized when you have a single green dye at the tip of the DNA. Okay? So even if your protein is not labeled, as the protein goes back and forth, there's this intensity fluctuation. And if you look at the periodicity of this intensity fluctuation to this fret fluctuation, it matched. So this told us that perhaps we can, this dye reports on its distance from the interacting protein, i.e. as the protein goes back and forth on this DNA, when the protein approaches the dye becomes much brighter, and then as it moves away, then the dye becomes dimmer. So we can basically make out the same motion of the protein on the single-stranded DNA. Okay. If you have questions, again, I'm briefly going over this PIFE method. So we named this PIFE, or PFA, or for protein-induced fluorescence enhancement. And the name has a definition that the protein actually enhances the fluorescence of a dye. So this is taken from another experiment, but this is a single molecule surface that we typically see on our computer screen. So you can see the intensity profiles of the substrate, the fluorophore attached on the substrate, and as you can see, we detect several hundred molecules per movie, per image. When you add protein to the substrate, you can already see that the fluorophores become much brighter. And this is, with naked eyes, you can see the molecules becoming much, much brighter. So we actually use this to see a binding and also a translocation of this antiviral protein called RIG-I. And this has been a mystery why this protein uses ATPase, what it uses ATPase for, but we showed that actually it moves around the viral RNA axis as it hydrolyzes ATP. So it is a motor protein, active motor protein that is able to translocate along viral RNA. So this was the first time that we used this method PIFE to publish any work. So this was early year 2009. And then, so you might be wondering about, what is the basis of this fluorescence enhancement? So according to literature, so this is a side 3 dye, and it undergoes interconversion from what's called a cis state to trans state, right? So, okay, so it can be in the ground state, cis state versus trans state. Only the trans state is the photoactive state. When it goes to cis state, it's inactive, which is depicted in this diagram. So what is thought to go on is that when the protein or some kind of viscosity factor approaches this molecule, then it limits its isomerization so that its average time that it spends in the trans state or the photoactive state is lengthened, and therefore more photons come out of that fluorophore, and there's an enhancement in signal. And also in this work, well, so then I'm skipping over this data, but this is a conclusion of our study, is that we can measure distance with this PIFE. So this is showing you the comparison between fret sensitivity versus this PIFE sensitivity. And as you can see, it regains sensitivity in the fret insensitive region, for example, below 3 nanometer region, because there's a very sharp drop in this distance range. And yeah, in this work, we did a lot of work, and we have shown that you need this 2-ring type of dye that is able to isomerize, right? So if you do the same experiment with a dye that's locked up, there's a molecule called Cy3b, which in between the 2 rings, there's this ring structure that lock up the 2 rings that it doesn't allow for the isomerization, then we don't see this effect at all. So, so that we published this work a couple of years ago. And for some entertaining, so this is a student, first author of this paper, and she actually wrote a rhyme of, I'm not a rapper, she's a rapper, and she's a dancer, so you can kind of imagine her dancing, and she actually presented her rhyme at like an international conference, so I, great. So I'm just gonna read it off, okay? So you all know about Fred, where two dice make a bet. When they're together, the red is bright, but apart, it is dark as night. When the protein moves back and forth, you can see fluctuating Fred, but pipe with just one die, changing intensity is what you get. So pipe is all you need, with just one label indeed. On DNA, it can be, which sets your protein free. So why Fred when you can pipe? Pretty cute, isn't it? So in fact, in my lab, almost everyone uses both Fred and pipe, because pipe is an easy, quick test when you want to test protein binding to a certain. Let's say that you have a protein that's known to bind to some kind of DNA, and you have like 10 different DNA substrates, and it's really difficult to label the protein. Then a quick check would be to prepare a slide, with 10 different DNAs in different chambers, and just apply protein, unlabeled protein, to see if there's enhancement or not. So it's a really quick assay to run, and it is not easy, because as you can see, Fred is a ratiometric assay where you then any kind of noise that affects fluorescence can be canceled out, because it is a ratiometric measurement. Pipe depends on a single fluorophore. So it is noisier, and it is a bit more difficult to take measurements, but it's doable. It's about the same, and exactly same setup and everything. So, okay, now I'm gonna go into the main story. I wanted to show you this Gangnam Style picture that we took last year. So yeah, we have some undergraduate students. The work that I'll show you was done by Peggy Chu, and in collaboration with Tim Lohman lab, and Edwin Anthony back then was a postdoctorate in the lab. Okay, so this is a brief overview of a homologous recombination. It's a DNA means of gene exchange between the two chromosomes, but it actually is a crucial mechanism by which DNA repairs itself. So homologous recombination does both the recombination itself, but also the repair. So we were interested in the repair aspect of it. So it is a very common process. It occurs in all life forms. And because of its importance, its dysfunction results in genetic diseases, such as many types of cancers, breast and prostate cancer included. And so this process has to be tightly regulated. You don't want to have it all the time, but you need to have it when it's needed. So there has to be means of down-regulating it, also means of up-regulating it. And so we know by a lot of literature evidence that when this process gets is disturbed at any step of the way, or any regulatory steps gets disturbed, then there are deleterious consequences. Okay, so this is a more zooming into what we actually wanted to do. So there is a recombinase. You probably heard about Rad51. And this protein is known to be an anti-recombinase. And you can already see why, as I alluded to, this is a protein that inhibits the recombination reaction. So they're kind of in a battle scene. But that's not everything about it, because one recruits the other, and there's some cooperativity between the two proteins too. So as you will see. So in simple terms, when there's a double-stranded DNA break, which is a big emergency in cellular life, what happens is that it gets, this top strand gets degraded by an exonuclease, and that process is called resection. Then when a single-stranded DNA is generated, 3'-tailed, Rad51 quickly comes and forms a filament along this single-stranded DNA. But what it also does is that it recruits this anti-recombinase called Srs2, and the function of Srs2 is to dismantle or disrupt this filament. Right? And then later other proteins come and lead it to further directions. So there were a lot of papers that showed this phenomenon. Our goal was to really zoom into this path, because there were questions about fundamental step sizes of this protein. How does it bind? How does it come in? How does it dismantle it? And what happens after the dismantling? And all these detailed mechanistic questions remained. And our tool is ideal for probing that kind of mechanistic details. So these two are the processes that I tell you about and what we've done to study the process. Okay, first we wanted to see the formation of Rad51 in real time. So here you have a fret construct, red dye here, green dye here, and we vary this length from 10 to 20. So as the filament binds, we expect to see the fret to go down, because due to the high flexibility of DNA, you expect to see high fret when the DNA is by itself. As the protein comes and binds, forms filament, it will become low fret because dye-to-dye distance will lengthen. Okay, so this is a fret histogram, which x-axis is a fret efficiency, and y-axis is actually number of molecules. So yeah, so this comes from something like 5,000 or more molecules. So it shows that when the DNA is by itself, it's high fret. So in gray is the DNA only, in orange is plus Rad51. So when we add Rad51 to this high fret DNA, we start seeing some fret shifting downward. But with such a short tail length, we don't see very much formation. As you lengthen the tail, which is this tail length, to longer and longer length, you can see that there's more and more transition into the low fret. Beyond 18 mer, we see a complete shift in fret from high fret to low fret, which tells us that nucleation or a stable filament formation requires about 18 nucleotide or above. Okay, so this agrees very well with its homologous protein called REC-A. So this was a satisfying result. Next, of course, the first data is just a snapshot of what happens after the filament forms, but we can look into single molecule traces. So these are representative traces taken from 10 mer, 13, and 15 nucleotide tail length. So everything starts from high fret, and you can see for 10 mer we see about 3 steps, fret stepping down. For 13 mer about 4 steps, 15 mer about 5 steps. And you know, because we're only showing a single trace per length as a means of showing more of these transitions, we can then obtain the fret values from before fret transition and after fret transition and plot it as a transition density as shown. So here what you're looking, x and the y-axis correspond to fret after transition and fret before transition. So here fret is going from 8 to 6 to 4 and below. So there's fret 8 transitioning to fret 0.6. Is that right? No, so sorry. Fret 0.8 transitioning to 0.6, 0.6 going to 0.4, and 0.4 going to something like 0.25. And then, so this represents the binding transitions, and that represents the reverse of it represents the dissociation. So this is a good way of showing many sets of data in one plot. And so basically what we see is 3 transitions, 4 transitions, and 5 transitions for 10, 13, and 15 mer, which tells us that assuming that one step represents a monomer binding of Rad51, one unit binds to 3 nucleotide, just based on the math. And this also agrees with some crystal structures for its homologous protein, that it is occupying about 3 nucleotide on the DNA. Okay. But not only do we see single steppings, we see some bigger steps. Of course, if there were 2 steps but we couldn't resolve it because of our time resolution, we cannot conclude that this is really one step. But as within our time resolution, we see some steps that seem to be oligomeric, meaning more than 1 unit. So based on the well-defined fret values that I've shown you in the previous slide, we can assign values of dimer, trimer binding to these. And then when we put this all into a transition density plot, it looks like, what is that game? Like Bejeweled game, everything falling from the sky. So it looks very crazy, but as we look closer, this is a monomer binding, dimer, trimer, tetramer, and then the second binding, a monomer, and third binding, fourth binding. And so this whole transition density plot can be reorganized as everything here representing the monomer binding, dimer, trimer, and so on and so forth. When we do a simple stat on this, we learn that majority of binding as well as dissociation occurs as a monomer or dimer. Does it make sense? Okay. So next question we had was the directionality. Does it bind from the junction toward the end of the tail, or does it bind from the end to the junction? So we couldn't use Fred for that because Fred just reports on the distance between the two dies, right? So we took out our weapon pipe, right? So we can, by positioning the die at the 3' tail versus 5' near the 5' tail, we should be able to tell if it's binding from one direction or the other. So they're exactly the same with the varying die positions, right? So when we did that assay, what we saw was that in this kind of construct, we see this stepwise increase in the intensity signal, which already tells us that it's binding from this end to that end. And then when we had this alternate substrate, we see a single step increase immediately after the protein addition, and then it plateaus at that level. So with this, and this is the average intensity taken from many molecules, there's slight difference in the enhancement because of the die position. And again, this is a detailed question. If you're interested, I can answer it later. So we do see that that directionality is really from the 5' direction to toward the 3' direction. Okay, so that's the end of the Rad51 story. Now let's look at the opponent, which is this protein anti-recombinase called Srs2. So these are yeast proteins that we also have human homologs. There's a human Rad51. There's no human Srs2, but there are other REQ family helicases that function similarly. Okay, so Srs2, which I'll represent in blue color, partly to wake you up if you're sleeping. So this protein is actually a helicase protein. So later on, I'll show you how this can also unwind the duplex part. But for this portion, we wanted to see what happens in the single-stranded DNA portion. So even without the Rad51, we wanted to test what this does on the single strand. So we use the exactly same DNA, the high Fred DNA of, I think in this case, I'm showing you a trace on the tail length T20. So when we added this protein, because it's the ATPase, we added the protein together with ATP. And very unexpectedly, we see this kind of very robust Fred fluctuation, right? It goes from high Fred to low Fred, like this. And so this is how we obtain a raw data. We look at single molecule, there's an intensity profile, Fred axis, and this gives us the Fred distribution for this single molecule, which you can see that there are two peaks. This is arising from the photobleaching. So there's this, somehow the protein makes the two die distance change in a very rapid manner in repetition, right? So that was very strange, but this was dependent on ATP. So without the ATP, okay, so in summary, the Srs2 somehow displays this repetitive motion on single-stranded DNA. Okay, so this was dependent on ATP. Without ATP, there's no fluctuation. Yeah? And
that was in the absence of Rad51.
That's, yeah, absence of Rad51. So let's look at Srs2 by itself first, right? So then one way that we test for the ATP dependence is by measuring this dwell time, right? Because one data provides maybe about 50 to 60 dwell time because it keeps on repeating. So if this, whatever is happening, if it's dependent on ATP, we should see it slowing down as we lower ATP concentration. So this is basically what you're seeing is a dwell time histogram at high ATP and low ATP. And we do see that, and we can, of course, take the inverse of this dwell time, which is the rate of this motion, and fit that to Michaelis-Menten kinetic, and we can generate this, sorry about this, should be lowercase, or Km and the Vmax for the reaction. Okay, so that's the ATP dependence. Another question, you may not come across this kind of question, but in the field of helicase, there's always a question about, is it a monomeric reaction or does it require dimer? So it is kind of a big issue. So we always want to know if it's a monomer-driven motion or if it's oligomeric or dimer. So we wanted to really nail it down. So we use this chemistry, which you guys may know very well. So we have on the slide, I've shown you how we cover PEG and biotin and their streptavidin. We got this biotinylated NTA from this Paul Hergenroth lab on campus in chemistry. So they actually synthesized it. Then we can charge it with nickel. Then it has a very high affinity to histidine tags that can be located on the protein. And this tag is used for protein purification. So we had it on the protein anyways. So then we can bind the proteins one by one. So now we have the unlabeled protein bound to surface, and we can add this same FRET construct, but non-biotinylated construct to this assay. And there it is. Yes, it's a monomer protein that is able to generate this motion. So I really like this assay because there's no fluorescent signal, right? Because the protein's not labeled. As soon as the DNA binds to the protein, we gain FRET, we gain fluorescence. Then we get to see this FRET fluctuation that is induced by a single monomer protein. So this was very satisfying. In fact, in many traces, what we see is these kind of bursts of fluctuation, and then protein or DNA dissociates, and then another DNA comes in, and then there's a burst of events. So we were able to see that this is a monomer-driven event. So to study further what's happening, what SRS is doing to induce this kind of FRET fluctuation, next thing we carried out was to vary the tail length, because we had all the substrates for the previous measurements anyway. And what we saw was that when the tail length is sufficiently short, like up to 25 or up to 20, so we tested, 13, we have every length, so we tested everything. So up to 20, the FRET signal is pretty similar. FRET fluctuation range is just about from 0.6 to about 0.8. But then it becomes slightly lower when you lengthen it to 25, sufficiently lower, and then it disappears when the tail length is too long. Okay. So whatever it's doing, the motion seems to be limited to a finite length of single-stranded DNA. And so we took the dwell times for all these lengths, and so these are tail length, and this is dwell time. You can see that there is a length dependence up to a certain level, and then there's a plateau, which really, again, tells us that it is acting on a finite length of DNA. So the picture that comes to our mind is that it is pulling the DNA strand in this kind of manner. And when the tail length is, and the length that is grabbed by the protein is finite, right? So when the two dyes are sufficiently close, we can read it as FRET change. FRET signal change, but when the dye is really far apart, then although it's doing the same thing, we don't get a readout because it's outside of the FRET sensitive range. Does that make sense? So to further test that idea, then we prepared 3 substrates of varying length. So we fixed the dye to dye distance to 15. And then we lengthen this from 15 to 25 to 35, right? So if it were to be length-dependent motion, then we should see some differences in signal. But in fact, they looked identical to each other, and their dwell times were very similar to each other, which again tells us that it is really acting on a finite length of DNA that is close to this duplex junction. We think is about 18 to 20 nucleotide distance that it is acting on. So we come up with this repetitive, we didn't know what to call it. So someone suggested that we should call it a scrunching, because it is scrunching up the finite portion of DNA over and over again. So we call this repetitive scrunching model of Srs2. So our emerging picture is that Srs2 binds DNA, translocates to the junction. By the way, this is really fast. It's beyond our time resolution to measure anything this fast. And so what we are monitoring is right here. When it gets to the junction, and it starts scrunching, letting go, scrunching, letting go in a repetitive manner. And this motion is dependent on ATP. So then the next question is this. Yeah?
So we learned that helicase is usually, like in the DNA replication process, is used to unwind like double-stranded DNA. So what prevents this helicase from going through the duplex region?
Hopefully you'll learn in the next few minutes why that is. So typically helicases, well, can I say typical? Yes, helicase is named helicase. Is it because it is capable of unwinding duplex? You would think so, right? But it's actually not. Helicase, there are a lot of helicases, more than 100 helicases. A molecule is called a helicase if it possesses this conserved domain. There are 7 amino acid motifs, motif 1 through 7. As long as a protein has that, it is called a helicase. And it turns out that many helicases that are named this way are not a good unwinder. But in this case, it is a pretty good unwinder. It depends on the concentration. You need to, in our case, we need to raise concentration to see unwinding. And what we see at low concentration, up to about 20 nanomolar, is this kind of scrunching. So which activity is relevant to in vivo? Maybe it does both. But we know in this context, we know that it's a monomer that acts as this anti-recombinase that was already kind of alluded to by previous studies. But we kind of nailed down that this is a monomer. This is the mechanistic activity of this protein. Okay, so it does all of these things, but is it useful? Is it really using this activity to contribute to its anti-recombinase function? So here comes the 2 players. Now we put both proteins together, again in the same DNA construct. So we form the Rad51 first. Then we add Srs2 to see what happens. So you will, because we preform it, it's already low fret. Then when we add Srs2, if it's dismantling it, we should see fret going up and whatever happens afterwards. And what we see is this. So there's a low fret because it's all filament is formed. As soon as we add Srs2, then there's this kind of gradual increase, and then there's this signature trace that tells us that Srs2 is scrunching, which means that it's completed its mission. It cleared all the filament, and it's sitting at the junction, and it's doing this scrunching. So that's nice. But what we see in majority of traces is not just this. This just means that whenever Srs2 sees Rad51, it clears everything and it does scrunching. But what we see in a lot of traces is like this. There is a filament formation followed by clearance, removal, then reformation of filament, and removal again. So this process occurs over and over again. Okay. So the conclusion we draw out from this is that Srs2 does, so it acts to prevent Rad51, right? Because it is sitting on it and it is grabbing onto the DNA, efficiently preventing the Rad51 from forming again. It does that, but only transiently, so it's not permanent, which makes sense because you want to have both this balance between formation and removal, so that when you really need it, then you can shift the reaction one way. And then when you need to disrupt it, then you can shift it to the other way. So we do see the exchange between formation and removal in a continuous manner. Okay, so to quantify this effect, we again use the dwell time because that's something very easy to do. So we just measure the time that corresponds to the formation, the low fret stage, and then the time of the removal, or the Srs2. So R for Rad51, S for Srs2, and then plotted it as tail length. What we plotted was the ratio between this R to S. So this ratio indicates the tendency for the formation, what is this word? Preference for the formation over removal, right? So what we see is that when the tail length is short, it is low, and as the tail length becomes longer, its preference for formation increases, or the stability of the Rad51 filament increases. And this is consistent with the nucleation size that we monitored before. Okay. So again, the monomer dimer question. Is monomer sufficient to remove the filament? So we use the same assay. We attached, immobilized the protein on surface by the same chemistry. And then we applied the preformed filament. Okay. And similarly as before, what we see is this appearance of fret and then removal process, right? Whatever is happening here is removing the filament. Then we have the signature that tells us that it's removed it. And so the conclusion we can draw from that is that monomer Srs2 is sufficient to remove this filament. And one nice thing about this is that we know exactly when the DNA, this entity binds, and how long it takes to remove the filament, right? And then the removal duration. So we collected all these removal times because we thought that it would be length dependent. And as we expected, it was length dependent up to this about 20 mer, and then it didn't change anymore. So this tells us that it takes sufficiently longer time to remove filament on a longer strand because it's more stable. Okay. And then now, this is the helicase activity, unwinding activity. So as I alluded to, this activity requires about 15 nanomolar protein concentration. So when you add that much protein, it unwinds. Okay. So that's what we did. Again, the same substrate. So in this case, we are expecting to see the loss of this strand if it completes unwinding, right? If it unzips the two strands, this should disappear because the other strand is the one that's biotinylated, right? So that remains bound, the other strand should be removed. So that's, so a nice thing about this assay is that we can just look at the green fluorescence over time and learn about the unwinding kinetics. And so this is showing the percent unwinding for different tail length. So as we expected, we see faster unwinding for longer length of protein. Probably it can harbor more than one protein. It can act in concert together. So this is an important part. So what happens then when the Rad51 is formed? Although I didn't have a chance to show you, it forms not only on single-stranded DNA, but also on duplex. So the hypothesis is that this may prevent unwinding. And that prediction was correct. So when you have Rad51 formed on single strand and duplex, unwinding slows down for all tail length. So we can see in this simplified manner that unwinding rate is tail length dependent when you do not have Rad51, but in the presence of Rad51, your unwinding rate is subsided. So I think, okay, so to see this more directly, then we can prepare this kind of substrate where two dyes are close to each other, then unwinding can be seen as fret decrease, right? So that's what we show here. When the Srs2 does it by itself, it's very quick, and fret decrease occurs immediately after the protein addition. But when there's Rad51, we see this long delay, right? Because it cannot get efficiently unzip the duplex due to the Rad51 holding its place in duplex. Okay, so that's all the data. These are dwell time analysis. And then the last bit of data is we use the same tethered protein assay, and we show that unwinding it cannot be occurring as a unit of monomer. A monomer cannot unwind duplex DNA. So what we are seeing here is that DNA binds in high fret, remains in high fret, and then it dissociates. Okay, so in summary, what we have learned in this study is that Rad51 forms from 5' to 3' direction, one monomer at a time. And then Srs2 is recruited, then it removes Rad51, and it goes into this repetitive scrunching mode. So here, what we learn is that it is an efficient anti-recombinase because not only does it remove the filament, but it stays stays at this position, and it repetitively scrunches the DNA, and thereby inhibiting the further formation of Rad51, right? But at the same time, what we also learn is that at the duplex, Rad51 actually occupies the duplex, and it prevents Srs2 from acting as a helicase, right? So there's this kind of twofold exquisite control. It controls each other so that Srs2 can act as an anti-recombinase, and this can prevent the duplex portion that does not need unwinding. So, okay, so this is the conclusion that I think I've covered everything because you guys need to leave soon. So this is the summary that Rad51 forms one monomer at a time. Nucleation requires 18 nucleotide, and it forms in this direction. Srs2 does this unexpected repetitive scrunching, and it is a motion to a finite length of DNA, and it is a monomer activity, and it uses that activity to prevent Rad51 reformation. However, it is only transient. And we also learned that the Rad51 formed on duplex suppresses the unwinding by Srs2. So these are the people that I already acknowledged, and these are the funding sources. And I can take more questions.
They don't usually clap for me.
Okay. Yeah, any questions? Yeah, wait.
So you mentioned that the Rad51 filament depends on the length of the single-stranded DNA that's available. So if there is like a stability sort of constant there, can you get sort of, so if you had a mixture of very short strands and very long strands, could the formation on a short strand be knocked off by a long strand that comes in? So can you get like a whole filament exchanged between DNA strands?
So this is inter-strand crossing. That hasn't been shown experimentally, but what kind of scenario are you thinking of?
Just so I guess it's not really physiologically relevant, but...
Okay, the long strand would have a very stable filament, right? But the short strand, the filament itself is, what happens is it forms and reforms, and it keeps on breaking itself because it's not stable, right? So I don't think once the Rad51 forms on the long duplex, it's going to lose it very easily. It does have some kind of low on-off rate, but it's very much lower than compared to the short strand. So, yeah. Yeah.
So you mentioned at the beginning this whole process is kicked off by your damage to the double-stranded DNA. Is the purpose of what you just described to keep that gene from being expressed anymore, or to make it possible for the DNA to get repaired?
Yeah. So this is. So for homologous recombination, when there's a DNA break by any source, that means that there is a damage to DNA, and so this is what happens. If it's a real DNA damage, Rad51 should form, and it should lead to the homologous recombination, the whole orchestra that happens afterwards. But there are cases where DNA, like foreign DNA or DNA, is seen as a damaged DNA, and so these kind of responses occur extensively and in unnecessary places, and then you need this kind of molecules to knock it off because it's not a true repair site. So it's a way of balancing and a way of checking itself to make sure that it only corrects the DNA that needs a repair. So
are there diseases that are related to non-correct regulation?
Yeah, they're actually, so I alluded to the human RECQ family helicases, which you may know Werner helicase, Werner syndrome, Bloom helicase causes Bloom syndrome. So these are different RECQ family helicases. If you have a deficiency or absence, then the consequence of that is a lot. Many different cancers. Cancer incidence is much higher. Lifespan is shortened. There are in the cells, you have a lot of DNA fusions, and yeah, many different, very deleterious consequences. So these proteins are crucial for this kind of, they're called genome caretakers, because they do this negative controls in these kind of processes. But in human, I mean, the yeast is nice because this is the only one. This is it. I mean, Srs2 is the one that knocks off But in human, there's still debates whether there are 5 different RECUs. So which one is it? Is it Bloom? Is it Warner? Is it RECU 3, 2, 1? So we're not sure which one it is, but they all coordinate to play this kind of role. So, any other questions? Yeah.
A question about the CY or Psi3B, saying that it doesn't have the chance to kind of change the isomer, that it won't have that effect. So even if it's basically trapped at a trans isomerism, it still won't have the same effect you're looking for.
So whether it's a trans, well, there is no trans in cis, I guess. So Psi3B is much brighter than Psi3. So it's good, but it doesn't have this, but it photo bleaches easily. So, well, experimentally, we just don't buy it. We bought it for this experiment. But because it doesn't, go through this inner conversion, then we don't see any enhancement. So, yeah. So.
Let's thank the person beyond the hand.